Hello, hello.
Hello, everyone.
So shall I start or are we waiting for someone?
Uh, Actually, we have right now 33 participants. So we'll wait for 2-3 minutes so that participant number reaches 250 or something. Then we can start. So first I'll introduce you guys. Then we'll give you instructions, the audience, the participants, and then we'll start.
Sure, thanks.
Good afternoon, everyone. Welcome back. We are now ready for our very first practical session on tracking of synaptic vesicles in vivo. For this practical session, our teaching assistants are Shravanti and Vidur, who are integrated PhD student in Dr. Sandhya Kaushika's lab, DBS DIFR Mumbai. Shravanti has done her bachelor's from Bhaskaracharya College of Applied Sciences, Delhi University, and now she is studying the multiple steps that allow synaptic vesicle proteins to exit the neuronal cell body in a motor-dependent manner. Vidur did BTech from Vellore Institute of Technology. He is now focused on whether ubiquitination alter the steady state distribution of the motor protein KIF1A. Now Malika will be giving the instructions for this session. Over to you, Malika.
Hi everyone. I hope all of the participants have downloaded the necessary files for this session and you have Fiji app installed on your system. And for this session, because this is a practical session, for any doubts or questions you can ask then and there. You can either raise your hand, you can put your question in the chat, or you can unmute yourself and ask. All three ways you can ask. And yeah, over to you, Shravanti and Vidur.
Okay, I'll start then. I'm going to talk about fluorescence recovery after photo bleaching. So for that, just give me a second. I'll share my screen. Okay, can everyone see my screen?
Yes.
Okay, thanks. Okay, so yeah, thanks a lot for the introduction. And what I'm going to talk about is, as I said, fluorescence recovery after photo bleaching. So the first question to ask is, what is it required for? Why is it needed? So the most obvious answer that most people use it for is to assess. There's a chat window in the middle. Yeah, the use case that most people use it for is to distinguish between two types of movement. One is a dynamic movement of molecules within the cell, and the second is where the molecules are stuck either because of interaction with certain complexes or with certain organelles. So to distinguish these two kinds of kinetics, which is a slow versus a fast state, is why people use FRAC. And how it is utilized? If you have a cell, this is a very simplistic cartoon of a cell, where you have some protein marked with the fluorescence marker. You can utilize a property of a fluorescence marker, which is the ability of it to transition to a non-fluorescing state, either a triplet state or a completely non-fluorescing state because of some oxidation or some reaction in the system. And this can be done by using a very high intensity laser, which keeps the fluorophore at a excited state for a long time so that a reaction can occur. So this leads to a complete loss of the fluorescence where the laser was shining. And over time, how fluorescence recovers from the steady state of the cell towards this bleach portion is what yields. Sorry, did someone speaking? Okay, I'll continue. So how molecules recover from the non-bleach region to the bleach region in region of interest is what FRAC essentially measures. And the difference between the recovery rates in these two portions is what FRAC tries to quantify. So, yeah. Now, there are multiple methods by which movement of molecules can be assessed. They are listed here by this wonderful review. The first one is fluorescence correlation spectroscopy, which is basically a method to uh, quantify the average residence time of any molecule. And the only limitation is it can detect molecules with a very rapid diffusion coefficient, which is 1 to 100 micrometers square per second. The second, which increases the width till which we can resolve the diffusion coefficient, is RICS, which is raster image correlation spectroscopy, which requires a CCD camera and basically uh, correlates the movement of molecules from one pixel to the second pixel across the camera. The third is the FRAC, which I'm going to talk about, which is based on a laser system and which requires bleaching of the fluorophore. And the other two are an NMR based, which loses out on spatial resolution and can only give you broad scale diffusion kinetics. And this is actually the technique which is utilized in brain scans because it's completely non-invasive, does not require any fluorophores, but it loses out on resolution and can only resolve up to 1 centimeter cube regions. And the final one is single particle tracking, where a fluorophore is required. And you have to have low enough concentration and low enough diffusion so that you can track single particles across frames. So the most utilized technique out of this, in biology at least, is FRAC. And this is the thing that I'm going to talk about today. If you have any questions about any other of these techniques, I have at least tried SCS and RICS and SPT. So if you have any questions, you can ask me later, but those are not something I'm going to talk about today. So moving on with FRAP, I already told you that FRAP tries to distinguish between these two kinetics, where you can have slow recovery, suggesting that the molecule is not freely moving or moving at a slower rate, versus another molecule which is freely diffusive in the cell. If you quantify the intensity in this region of interest over time, what you can have is two different curves. In the top one, which is highlighted by light green, you can have a fast recovery. So, and in the dark green one, you can have a slow recovery. That is one parameter that distinguishes the two, let's say, wild type versus mutant, or two different proteins. The other parameter that can be looked at is, after an extended amount of time, how much intensity does the fluorescence recover by? So as can be seen here, by the time this graph starts to saturate, green has, let's say, recovered by around 70 to 80%, but the dark green only recovers to 20 to 30%, which suggests that a lot of the molecules that are present within the cell are not capable of coming back to this particular compartment, either because it is bound by a membrane, or it could be a membraneless organelle, or it could be certain complexes that lead to its uh, movement, that impede its movement. So, yeah, going on. So to quantify this, what we do is to model these recovery kinetics using a single exponential. I'm not going to go into the derivation of the single exponential. I've put up a link over here. If anyone is interested, they can follow this link, and it wonderfully explains why this single exponential is used and what all parameters can be acquired from it. However, points to note from this single exponential is that it has two terms that are extracted from this recovery profile. The first is tau, and the second is A. What A denotes is the fraction up to which the fluorescence recovers post photobleaching, which is termed as the mobile fraction. This can be imagined as the percentage of molecules that can freely diffuse within the cell and which aren't bound to anything inside. And the immobile fraction is the one which is bound to something and cannot freely diffuse within the cell. The second thing is the half time called T half, which is related to this tau function by this formula, which is ln 2 by tau. And this parameter basically tells you how long it takes for the fluorescence to recover half of its maximal value, uh, So these are the two basic parameters that one can gauge from recovery profiles. Now, this assumes that there is only one fluorophore of interest, and it does not have any complex dynamics such as two diffusion coefficients or two states or any of that. This is a very simple model where one protein can be assessed across two different conditions, let's say a wild type and a mutant condition. So there are other uses of FRAC apart from quantifying the diffusion rates itself. Certain use cases is what I've listed here. The first is to detect compartments within the cell, such as let's say if a protein is bound to organelle, a membrane-bound organelle, or whether it is inside that organelle. So if it is inside that organelle, the fluorescence within the organelle will never recover. However, if it is just peripherally bound to the organelle, it may recover over time. The second can be to detect interaction between molecules, which is where you have two different fluorophores tagged to two different proteins. And if you do a frac on one of them with the second one constant, you can assess the recovery of one with respect to the second, thus suggesting an interaction between the two molecules. And the third one, which I generally use for my experiments, is to characterize asymmetric transport of molecules within the cell, in our case, a neuron. So here, when I photobleach a particular region, you can have recovery from the left or the right. The difference between recovery from the left or the right leads to an asymmetric transport and something which can be quantified. So there are certain practical considerations that have to be paid attention to while doing this experiment. The first is that this half-life rate fitting is only a pragmatic method. It does not lead to any information about physically relevant parameters. And what I mean by physically relevant parameters is things like its actual diffusion coefficient or whether there are compartments or not. This is just an experiment which can, um, uh, which suggests a thing with certain assumptions that have to be confirmed by other experiments. So uh, on the bottom here, I've listed a few of the uh, things that the half time can depend on. It can depend on the hydrodynamic radius of the molecule of interest. So as an example, if a protein is unfolded versus if it is tightly folded, the tightly folded version will have a higher diffusion coefficient because it's much smaller. And thus it has a less a hydrodynamic radius. The second is viscosity of the environment, which can be perturbed by multiple things, such as, let's say, the temperature, the amount of heat shock proteins within the cell, the age of the cell has been shown to be correlated with viscosity. So all of these factors also lead to changes in viscosity of the environment. The third is compartmentalization in the cell, and as I told, the cell is non-homogeneous and there are multiple compartments. So one has to really take care of the regions that they are bleaching in. And if there is any difference between wild type and mutant, uh, uh, like differentiating between all of these parameters becomes very difficult just based on FRAC. So there are other experiments that are needed. The fourth one is a crucial one in experimental paradigms, where we generally consider a cell to be static, but cells are rarely so. They will move, they will change their area, they will change their volume, they will try to move from one place to the next over time. So these dynamic movements leads to complicated regions of interest which can be bleached, and thus it becomes very difficult to uh, delineate whether it's the dynamic movement of the cell which is responsible for recovery of the fluorophores. And the last two are experimental, which is basically the first one, which is blinking of a fluorophore, which is the tendency of fluorophores to transition to a dark state for a small amount of time, around a second or two, which is called a excited burst. And that is the shape of the bleaching area. So one really has to consider these two events. And for these two, what you have to do is you have to have the same bleach shape in all of your movies as well as the area. So if you go with the circle, you have to maintain a circle with the same radius every time. And if you go with a square, it should be a square with the same dimensions every time. And the second thing to prevent blinking of a fluorophore is to have similar levels of photo bleaching. So if you're photo bleaching it to, let's say, 80%, you should make sure both wild type and mutant are bleached to 80% of the initial values. The second set of practical considerations that have to be kept in mind are bleaching. So the first one is that the frame rate should be at least as fast as half the half-life. So just to give you a sense of what this means, I'll just annotate. So here you see an intensity profile over time. So you have to have some sense of what half-time you're going to expect from your experiment. If your, let's say, frame rate, so let's say this is your 0 second, let's say this is over time, this is 5 seconds. If your first frame is at 1 second, you've already crossed the half-life and thus you do not know what baseline you're starting from. Since you don't know what baseline you're starting from, the calculation of T half as well as the mobile fraction will be completely skewed, because now you started from a region which is much above the of post photo bleaching region. So your frame rate should at least capture 2 time points before your T half, so that you can have an accurate estimate of where the post recovery profile started from. So that is the first thing to keep in mind while doing this experiment. 1 second, I'll just, okay, yeah. The second thing that is to be kept in mind is that photo bleaching has to be optimized to around 80% of pre-bleach levels. Uh, you can have higher or lower amount of photo bleaching, but generally what that does, if you have a lower amount of photo bleaching, again, because of the noise in the system or depending on the camera that you use, the recovery may not be mapped out correctly, and we'll get to it when we're analyzing the uh, movies that are to be covered in the session just after this. So you'll see that if noise gets too much over time, if you have only, let's say, 30% photo bleaching, you cannot directly assess the recovery because of the noise. So you have to have sufficient amount of photo bleaching such that you can assess the recovery with a narrow standard deviation. The alternate, which is to have a higher amount of photo bleaching. Let's say if you do 100% photo bleaching, the caveat of that is that the cell that you're interested in is undergoing a lot of stress because of the high intensity of light that is being exposed to it. So no matter what lasers you use, there will be certain amount of heat generated in the system, as well as reactive oxygen species, both of which can damage the cell. So I'm going to take the example of GFP, where you generally excited by a 488 nanometer wavelength. There you can, if you bleach it by 100 or even greater percentage, what you can have is over time, the let's say the cytoskeletal elements or the structure of the cell that you're interested in will start getting disrupted, and you will see that in your movies. It depends on what system you're using. So I'm not going to show our neurons case, because it's very different from systems to system, and that's something that has to be standardized based on the system that you're using. As an example, if you have HeLa cells, they will start blebbing over time. If you have primary neuronal cultures, they will show reduced amount of neurites that they throw over time. So this damage is something that has to be observed based on the system that you are using. Uh, Furthermore, if you are interested in certain compartments of the cell or certain cytoskeletal elements of the cell, what you have to ensure is that okay, there's a question. I will take all questions after I finish the talk, just because just to keep the flow going. So yeah, if you're doing any sort of membraneless organelle or membrane-bound organelle or cytoskeletal element imaging, you have to ensure that the post-bleach recovery, when this recovery has completed, looks almost exactly the same as the pre-bleach recovery. That's the most important thing over here. If it does not look the same as the pre-bleach level, and there is something wrong, and you have to reduce the either the time that the laser shines or the laser power for which the laser shines. The third is that acquisition laser power should not lead to greater than 50% bleaching over the course of the experiment, which is, if you have this intensity profile, if you don't do photo bleaching, just based on the fact that there is laser that you're shining or lamp power which is shining on the cell, you will have certain acquisition photo bleaching occurring within the cell, because of which the intensity reduces over time. This intensity should not reduce greater than 50%, because if it reduces greater than 50%, your recovery profile, which, let's say, if in an ideal condition it goes up to 80%, it will not go up to 80%, it will only come up to 40%, let's say. There are ways to counteract that, but they're not always error proof. And thus, it's best avoided to have intensity uh, bleaching over the time course of your experiment to be above 50% intensity. And the last is that ideally, in such a case, since acquisition bleaching you cannot prevent and will happen at some rate, ideally you should have an independent region or independent cell that is not cytosolically connected to your region of interest, such that you can monitor acquisition bleaching in that cell and correct for that acquisition bleaching in your photo bleached cell. So those are some things that are to be kept in mind while doing the experiment. I am not going to talk about the ways you actually uh, tune the microscope such that your image is not saturated and such that you fill up the gray value, because I saw yesterday's lecture and they did a quite job explaining all of that. So the final thing that I'm going to talk about before we go on to the tutorial section is the selection of fluorophores, which is a step prior to imaging of your experiment. So when you're choosing the fluorophore to be photobleached along with your protein of interest, you have to ensure that the fluorophore is photostable. And it's not a fluorophore that, as I previously said, just loses intensity over time just because of acquisition bleaching. So that's what it generally looks like. Generally, GFP and these fluorophores come in this category. And old generation fluorophores like M-Cherry do a pretty bad job and they photobleach quite rapidly. The second is that the fluorophore should be able to photobleach in your microscope setup. So depending on the kind of microscope you have, or depending on the power of the lasers that you have, you may have an overexpression system where you will have to bleach the region of interest for at least 10 seconds to have any loss of fluorescence. So that then implies that for the fluorophore that you chose was the wrong fluorophore, such as, let's say, quantum dots are well known that they undergo very minimal photobleaching. So quantum dots will not be a good choice for FRAP-based experiments because you won't be able to photobleach them itself. And the last thing is that the fluorophore should not alter the protein of interest itself. So this can be by two ways. The first is by M-Cherry, which is the most uh, uh, like simplest to see, where in this neuron of interest, we can see that there are compartment-like aggregates that form in a completely cytosolic M-Cherry. This is not labeled with anything. This is just soluble M-Cherry and it still compartmentalizes in these puncta-like things. Tag it's not the greatest choice to assess FRAP kinetics because you know that some of it will be bound as aggregates. So you have to choose one that does not seem to aggregate. GFP seems to be the best in all scenarios. The second is that the fluorophore of interest should not change the size or interaction of the protein of interest. So this is ensured by tagging the fluorophore at both the N as well as the C terminal and seeing which one best phenocopies your wild type protein localization in either organellar localization or compartmentalized localization. And the last thing, it shouldn't be such a big size that it completely alters the kinetics of movement of the protein of interest. So if you have a small protein, you have to have a small fluorophore attached to it. You can't have a huge fluorophore attached to it. So as an example, there are newer fluorophore based on what's it called? You have tandemization of fluorophores where you can have 7, 8 fluorophores to have a super signal GFP. If your protein of interest itself is just 20 kilodalton, you can't have 60 kilodalton worth of 6 and 7 GFPs stacked to it. Because then when you do FRAP, you're not assessing the protein's kinetics, you're essentially assessing the 6x GFP tagged proteins kinetics. So that's something that one has to keep in mind. So with this, I'll move on to the practicals, but before that, I'll like to address any questions that participants have with the theory of FRAP. So the first question that I can see is that what kind of fluorophore considering blinking kinetics is suitable for FRAP experiment, and also is it dependent on quantum yield of the fluorophore? Okay, so I guess this is this slide. What kind of fluorophore? That depends on your FRAP experiment. So if you're looking at very rapid kinetics, let's say within the second time scale, you have to be sure. So GFP does a brilliant job. M-Cherry does undergo a lot of blinking kinetics. So at very small time scales, M-Cherry may not do a good job. M-Scarlet does a good job at very small time scales. If your time scale is much larger, let's say in seconds, around 10 to 20 seconds, then you can sort of average out the blinking effect of fluorophore. So it's not that big a deal if your kinetics are slow. The second, which is, does it depend on quantum yield of the fluorophore? It does, however, as I told you in the second part of this slide, photo stability is one thing. Quantum yield, the only thing that quantum yield will change in this particular experiment is the SNR, which is the signal to noise ratio. For all experiments, higher the signal, higher will be your confidence in the experiment. So it does depend on the quantum yield of the fluorophore. However, in case of FRAP, it you can always like uh, you can always increase the laser power, or let's say express a bit more protein such that you can have sufficient fluorescence to assess from. Now, overexpression of protein is another fact that has to be looked at later, which has to be seen whether the overexpression yields similar kinetics as the wild type protein. But that is always an option with the FRAP experiment if overexpression of protein is not an issue. Okay. So the second question is, I was unable to achieve 80% bleaching for GFP tag protein. Is it something to do with bleaching parameters? Okay, so for this, this depends on your experiment of choice. So as and your system of choice. So if you're using, let's say, HeLa cells, you will have to decrease the transfection amount so that your GFP overexpression is lower, so that you can come up to that 80% threshold. That is one. The second thing that is to be looked at is um, whether there is autofluorescence or noise in the system. So sometimes it's not possible to go down to 100% or even 80%, because no matter how much you photo bleach, the lowest intensity you are getting is not from your fluorophore of interest, but it's from some background autofluorescence, some organelle, or something on the cover slip itself, creating noise in the system. So that's something to be taken in account if you're not able to come up to 80%, even after reducing the concentration of GFP in the system. If you take care of all of these and you still have only, let's say, 40 or 60% bleaching, uh, then 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 that's a caveat, and you really can't uh, prevent the system from doing that. So you'll have to continue with your experiment. But it really depends on the system that you're using. So the two ways by which you get 80% bleaching is either by lowering the GFP amount, or by in, increasing the laser power. So I hope that answers your question, Shalini. You can uh, maybe later we can go into a breakout room where you can explain your exact paradigm, where we can go over this further. The third question is, you said that the post-bleach intensity level should be similar to pre-bleach values, but would that not depend on the mobility of protein of interest and vary? Please clarify this point. Okay, so this is for this slide. So in an ideal scenario, uh, where the protein is completely cytosolic and there is no immobile fraction, the pre-bleach value should be almost equal to the post-bleach values. But that's an ideal scenario that considers that acquisition bleaching is not happening. There is no immobile fraction. So that is rarely going to happen in your experiment. You will have some loss in fluorescence over time, because one, you photo bleached a region and therefore some fluorescence is lost, and second, there is acquisition bleaching in the system. So the exact intensity post bleach should not be equal to the pre bleach value, but the post bleach, um, what's it called? The post bleach, like if you're assessing some organelle or some compartmentalization, that static distribution should come back to normal, is what I meant. The exact intensity may or may not come back, because experiments will always have some amount of photo bleaching. But you should always see that the structures that you just photo bleach, those structures should come back to wild type like states. That's what I meant. And because that ensures that there was no damage to the cells, and whatever you are monitoring is because of the dynamicity of the object that you are interested in. I hope I clarified it, Anubhav. Then the fourth question is, can you please elaborate how using FRAP you check asymmetric transport of molecules of your interest? Okay. I'll just go back to the movie for this. So, generally, when most people are bleaching a cell, they will have a three-dimensional cell. And because generally you do photo bleaching on a point scanning confocal, you have a pinhole which reduces uh, the Z in which you're imaging. So inherently you will be, let's say, assessing kinetics only in a two-dimensional front, with the third dimension being more or less much slower or 3 times slower than the X and Y axis. So you can neglect the Z axis. So what you have is something which is from left, right, up and down. However, in my case, since I'm using a neuron is a long neuron throws a process which is long and slender, and this neural process is just at the resolution limit. So inherently, when you photo bleach this neural process, you don't have any up or down recovery. You will have only left and right recovery. So what this allows us to do is to assess recovery from the left and right, and to assess if recovery from both of these regions are equivalent. If there is any asymmetry in the recovery from the left or the right, you can then modulate using fixed second law and assess how that asymmetry is being brought about within the cell. This can also be uh, done similarly in the cell. However, it becomes much more complicated because now you don't only have left and right, you also have up and down. And you have all combinations of up, left, bottom, right, top, right, and bottom, left. So just within a three-dimensional cell, assessing asymmetric transport then becomes very difficult. So you have to start with the assumption that there is no up and down recovery, and there is only 1 degree of freedom within the cell, and that's how you can assess asymmetric transport. I hope I answered your question, Alice. And if anyone has any further questions, I don't see any more. Oh, okay. There's another question by Tejil. What is optimal transfection efficiency required to achieve 80% bleaching? So that I can't really tell about each particular system. That is something that will be done by trial and error. So you'll have to like transfect once, see if you can. It's not such a high overexpression that you can't bleach 80%, and then iterate towards that amount of fluorescence so that you can optimally achieve 80% photo. And 80% is ideal. However, anything from 60 to 85% is doable. So that's something that everyone will have to experimentally uh, iterate and standardize. Okay,
Hello, Vidur.
so yes.
Yeah, there is one question on YouTube Live. It says, "Does the dynamics of vesicular structure can the dynamics of vesicular structure be assayed by FRAP, and cerulean blue can be used as a fluorophore or not?"
Okay, so I don't know about cerulean blue. We can always check out on FP base, which is a resource for all fluorophores. So one can always use that website to check whether a particular fluorophore can be used for FRAP effectively or not. And the second thing, which is, could you repeat the second question, which is whether we can assess organelle dynamics based by FRAP? Is that the question?
It's can dynamics of vesicular structure be assayed by FRAP?
Okay, so What I understand from dynamics of vesicular structure, one is if the vesicle is larger than the diffraction limit of light, such that you can monitor two different regions. Let's say things like the ER. In the ER, people have done localized photo bleaching and assessed recovery within ER as well as dynamics of ER over time. The second part, which is if the organelle is sub-resolution and you cannot resolve it, then you can't assess dynamics of that organelle. But what you can assess is recovery or interaction of any protein with that organelle. So let's say if a protein is bound to that particular organelle, based on photo bleaching, you can see how much of it recovers over time, and thus you can conclude how dynamic a protein is on a organelle. I hope that answers the question. Okay, I'll move on to the next question till then. Which is, will two interacting proteins have similar kinetics of recovery? Is that the rationale behind studying?
Vidur, can you hear me?
Yes, Sandhya. Okay, I lost you.
Vidur, can you hear me now?
Yes, I can hear you.
Okay, so for whoever asked that question, you can look at a recent paper from our group in the first author, Sudeep Mondal, the same person, microfluidics, where we bleach a non-moving mitochondria and look at a moving mitochondria to see how much it contributes to the non-moving mitochondria. And we show movies where you can see events where a mitochondria will give some contribution to a stationary mitochondria and yet continue ahead. So there will be recovery in the time scale of a movie, and that example is there. But the other things all hold what Vidur said, but there is this one or two cases where you can look at what the contribution is if the objects are discrete and far apart. Otherwise, what you can look at is just movement of proteins in a tubular network-like structure, which is what people do for ER and mitochondria, as you mentioned.
Yeah, thank you, ma'am.
Yeah. So the last question that I can see is, will two interacting proteins have similar kinetics of recovery? And is that the rationale behind studying interactions via FRAC? So yes, that's what I was implying. If, let's say, if a protein is part of a complex and mediated by protein-protein interactions, so you can always photo bleach one of those components and monitor its recovery in that complex. And that's how you can study interaction within the protein. The second thing is what people have done is used FCS to actually study two interaction between two proteins which are completely cytosolic and not uh, um, part of any complex, like any part of a bigger complex apart from those two proteins, because FCS leads to a higher resolution at that faster diffusion uh, uh, time zones. So for just two proteins, people use FCS. If two proteins are part of a larger complex, you can use FRAC. And I can always go into FCS later if anyone is interested. Okay, so I guess that's the end of the questions. We can move on to the tutorial part. If anyone has any questions, we can always use the breakout room to answer that. So I will, since everyone can open Fiji as well as the tutorial dataset, or the FRAC tutorial dataset that I had circulated, that would be great. I'll go step by step and I'll wait every major step for around 10 to 15 seconds. If anyone has any question, I don't know if raise your hand option is here or not, but you can either type in the chat or do something like raise your hands and I will answer that. or just unmute, whatever
Yeah,
works.
I think raise your hand option is there.
Okay. Then if the co-host can just pause me and or escort me to a breakout room, I can answer the question there.
Yeah, sure.
Yeah. Okay. So I am going to share my screen. Everyone, please open Fiji and some spreadsheet manager like Excel or Google Sheets. Anything will do. Okay. Can my screen Fiji's window as well as the test data sets?
Yes.
Okay. So everyone can open um, uh, everyone can open the test sample 1 as well as the no bleach control and have them side by side. So the moment you open these test data sets, I've already marked a region of interest. However, you can always use this line command to mark your own region of interest. You just have to right click this line, go to segment line and mark whatever region of interest you want. So it's something like this. Very vaguely. I'm not. So generally what we do is using plus. So if you press the plus button, you can zoom into an image. And what you do is just click on particular regions of the neuron or whatever regions you want and select this region. And if you double click the line button, you can have a line width option. So you just go up and select a line width that you're comfortable with. Let's say 5 in this case. So whenever you want to save an ROI, you just press T, and if you press T, that ROI will now be saved in your ROI manager.
Hello,
And you can
Vidur.
always, yes.
Khushbu has a question.
Oh, yes.
Khushbu, you can unmute and ask.
Vidur, I'm not able to find which file. I think I'm confused.
Oh, okay, okay. You
Hello?
downloaded the zip file, right?
Yes. The name, the one
Which
which
is
is named for imaging workshop, right?
no. This is the frap underscore tutorial.
Oh, yes.
You've opened that?
I'm not able to open that.
Oh, are you using anything apart from Windows?
No.
Okay, so do you have WinRAR or any like
Yeah. I'll try to
you
open
have
with
WinRAR?
that.
Just try with that. You'll need to unzip it or just right click. Generally, Windows can unzip it as well.
Okay.
Yeah. Just let me know once you're done.
Yeah, I'm done.
Okay. So you can go on to the sample 1 folder. And there you'll see there's one test sample 1 and one no bleach control.
Hi Vidur. So I am getting an
Yes.
.rar file. So I'm not getting a zip
So
file.
that .rar is zip. If you have WinRAR, what you need to do is just right click and unzip it in whatever location you saved.
It's not a zip file. It's not getting extracted.
It's not getting extracted.
No.
It's a I mean, what software did you use to open it? Did you try WinRAR or what are you using?
Hello, Khushbu. We are sending you the extracted files.
Yeah, okay.
Yeah, I
Thank
think
you.
that will be best.
Yeah.
Yeah, and I think some Mac people are having a problem. Someone messaged. Okay, I'll just wait for a second. When people have like unzipped it or like extracted the files, just let me know.
I got it. Thank you, Vidhi.
Yeah, got it. Okay. Has everyone got it? If just unmute yourself and tell me now, or otherwise I'll
Receive
proceed.
it.
Sorry.
I'm yet to receive it.
Oh, okay, okay. Yeah.
Hello, Khushbu, we have sent it. It will come in 2-3 minutes.
Thank you.
Yeah, we have sent to all the participants the extracted files.
Okay. Let me know once you've opened it. It's sample one, sample one, and no bleach control. Oh no, a sample. Someone put it. Someone had some question. I have sample one, sample two, and equation file. Is there any? No, that's all. Just these three. Okay, has everyone downloaded it? Okay, I guess I'll continue then. I guess everyone has it now. If anyone has any problems, just unmute and speak.
Yeah, Vidhur, I think you can continue. Most of the people got it.
Okay, sure. Okay. So now you can open the file. I'll continue from the beginning. Just open Fiji and open no bleach control and test sample by dragging these images into Fiji. It will directly open the files. So now what you'll see is that I have already marked an ROI of interest. If so generally, since everyone is not going to focus on neurons, in this case, since it's a neuron, we use a line because this is a one-dimensional process. So you can just monitor recovery in this narrow line. However, the region of interest that you're going to mark over here is ideally going to be the shape of the photo bleaching that you used in your microscope. So what you have to do, and this depends on the microscope, but what you have to do is you have to export the ROI file, which is a region of interest file, from your microscope, and that generally gets saved in your metadata for the images, but it may not always get saved to the metadata, in which case you'll have to export that ROI file separately. So that's something that really depends on the manufacturer of the scope. I know for one, which is Zeiss scopes, will have ROIs that get attached with the metadata. So if you directly open it in Fiji, Fiji can directly read those ROI files, and those ROIs are something that you can use for monitoring recovery directly. So you don't have to draw all this. However, there are other scopes such as Olympus scopes, which may not necessarily export the ROI along with the images, and thus you'll have to export them separately. So that's something that has to be taken into account while you are imaging. So if you have the ROI files, well and good, you don't have to do all of this procedure. If you don't have the ROI files, what you have to do is you have to shape the location of the photo bleaching that you did. So in my case, if you play this green movie, what you'll see is that I did photo bleaching in this one region, which is marked by this yellow line. And thus, I already have an ROI file for this. So just as an example, what I'm going to show you is how to make an ROI and save that ROI in Fiji. So in our case, if you open the neuron and you just hit the plus button in your laptop, you can zoom into the image and you just have to mark different points on the neuronal process such that that line is drawn. To specify the line width, which is how big this line is, you double click this line icon, which will open the line width column here. And here you can specify whatever width you want. So for our case, the neuron generally encompasses around 3 pixels. So I'm going to put a line width of 3. And to save the ROI file, you just have to press T. So if you just press T on your keyboard, the ROI file will come directly. And to save it, you just select it, you press more and press save, and it will give you the option to save it. Is that okay? I guess everyone has followed till now. So now the second step, once you have marked your ROI, is to basically get the intensity value over time. As you can see, this is a movie. So there is a slider on the bottom. If you press the play button, it's basically a movie which will continue over time. So I'm going to start with the no bleach control, which is basically just imaging the neuron without any high intensity of laser. It just leads to acquisition bleaching, which is what I was talking about before. And that acquisition bleaching, as I said, shouldn't be greater than 50% over the duration of your. So right now, what we're going to do is assess what level of photo bleaching is occurring without any intentional photo bleaching. So for this, once you have the ROI marked, you have to press image on your Fiji window. You have to go to stacks and then you have to press plot z-axis profile. I will wait for about 15 seconds so that everyone comes to the same stage. Here, I'll just talk about what this is. This is basically just the intensity on the y-axis, which is plotted over time, which is the x-axis. So natively, what Fiji does is to scale this window towards the maximum and minimum values of both time and intensity. However, what we want to see is that what percentage of photo bleaching is occurring. So for that, we'll have to resize this window, and you can simply double click the y-axis, which will bring forth scaling that is required. So just for 0 to 1072, and now you can see that the photo bleaching is less than 30% over the entire time.
Hello, Vidur.
There are certain questions. Yes.
Yeah, there are
Yes,
two questions
Malika.
in the chat. Can you see?
Yeah.
Yeah.
Yeah, I can see that. I'll just address them. Can you please repeat after clicking on the image option? Okay. So you have to press image, then you have to press tags, and after tags you press plot z-axis profile. This is the 3-step thing. And the second question is, would please show once again how you generated the plot? Oh yes, that's the thing that I just showed. I'll just pause over there so that others can follow. So it's here. That's plot z-axis. Yeah, okay. So I guess everyone has come to this stage now. So our main aim was to see how much photo bleaching is happening over time. So here we can see that whatever conditions I use, the photo bleaching is less than 30%. If it is greater than 50%, what we start seeing is non-linear kinetics, which is the acquisition photo bleaching starts becoming like an exponential decay. And that's difficult. So for now, we're still in the linear range, which is good. What we do is to press this list button, which will list the values as well as the frames for all of the movement. So this is the final list value. So what you do is you just copy paste this list by pressing Control C or Control X, or if it's Mac, then Mac C and Mac X, and just paste it in Excel or any spreadsheet software. And here, if you just plot this, what you can see, just use a better plot, 1 second. Yeah. How did you come to the values? Okay, 1 second, I'll just go back. Here, when you have this plot, you just have to press list, which is the bottom left button. And once you press list, you just copy paste these values into Excel and plot it. Once you plot it, what you can do is you can add a trend line. Or second, how did you change the values? Zero thing. Okay, 1 second. Here, what you do is you just double click on the y-axis. So once you double click on the y-axis, so if you just hover over the y-axis, you'll see that it'll brighten up to orange. You just double click it and you will see this column where you can rescale your plot to any number that you want. So here you can just type 0 and the maximum you can just let it be, because what we're interested in is how far this is from 0, how we normalize. Okay, so going on, and why is it required? Why it's required is to just check whether your acquisition photo bleaching is greater than 50% or not. So this is a quick and dirty way to check if whatever you're imaging is within bounds for FRAP. You can quickly do it maybe on the microscope itself when you're assessing the laser power, the exposure time and all that. And you settle on a laser power, exposure time, pixel density such that the photo bleaching is not greater than 50%. Generally, practically, you will get anywhere from 10 to 40%. However, in certain cases where your fluorophore intensity is very low, you may also go down to 70 or 80%. That's why. So it's a quick and dirty way to check the settings are okay. Why we are setting lower value to 0? I didn't quite understand this question. Oh, you mean why are we rescaling this from 0 to 1000? Because if it was not scaled, so generally how it starts out is like this, from 1000 to 700. So just looking at this graph, so right now, since the value is 1000, it may be easier to see that you just divide it by 10 and that's your acquisition bleaching. However, you can have random numbers like 3556 or 4356, and you can have some other random number here. So it may not always be the best idea to leave this as auto scaling. And in that scenario, it's a good idea to just go here, press 0, and the 50% mark will always be marked by a line. So you can quickly see whether it's less than 50% or more than 50%. Yeah. So that's why it's a good idea to always do this at the microscope itself. Okay. So once we have this, what we do is a very simple thing, which is just to calculate the slope of this line. The slope of this line can be calculated by two ways. One is a very simple formula where you just write is equal to slope, and in the bracket you have your known y's, which is your intensity, and after a comma you can put your known x, which is your time, and you have a slope, which is in this case minus 1.385. Oh, sorry. One thing I forgot to mention before you calculate the slope, what you have to do is you have to normalize these intensities from 0 to 6. Press equal to, press the first row of the intensity divided by, again, the first row of intensity. The only change that you do is you put a dollar sign before and after the B. So it's $B, $1 if you're using Excel. So the first intensity that is marked will automatically come as 1. And if you just double click the bottom right of the cell, it will normalize the entire intensity value, as you can see, from 0 to 1. And if you just plot this intensity, which is normalized from 0 to 1 with time, now you can get a slope which is normalized from 0 to 1. So in this case, let's say it's minus 0.00131, which means that every one frame you are losing approximately 0.00131 intensity per unit intensity that you quantify. So as an example, On average, this pixel intensity, this frame intensity is this minus this. And you keep subtracting as you go lower. This is what it means. Just so that everyone is on the same page and everyone can visualize what I'm doing, I'm also going to do it on this chart because since it's visual, it may be easier to understand. So what you have to do is just click on Trendline. Will you please repeat how to calculate the slope here? Yes, yes, of course. So what you do, once you've normalized your intensities, you just have to put equal to slope. And in Excel, this is a wonderful thing where it will take all of your Y intensities, your normalized Y intensities. So it also gives you hints of what is needed there. So here it's asking us to put some known Ys. So we will put this entire column C, which is our normalized intensity. Then put a comma. And now it's asking for known X's, which is our time. And we'll put that and just press enter. So this will give us a slope. And just to show what this equation is doing, I'm also going to do it on the chart. Now this depends on the version of Excel that you have, but something similar will be there in every Excel starting from 98. So what you do is you click the trend line option and go to more options from the trend line option here. And once you go to the more option, uh, once you go to the more option, here what you can do is equation chart. And as you can see, this equation is basically the formula to map out this slope, which is minus 0.0013x plus 0.9878. 0.9878 is irrelevant because that suggests the intensity at which we start. And since we don't care, we've already normalized it to 1. It will more or less be close to 1 depending on the noise in the system that you have. What we care about is this slope thing, which is minus 0.0013, which is what we calculated from the slope function. So this is a more intuitive way to do it. I hope everyone has followed till now. Our test sample now. This is all that needed to be done from the no bleach section. The only thing, so if one thing to note here, since the is that since the intensity or reduction was very small, only around 20 to 30%, we were still in the linear range. And that's why we could model this with a single line, a linear line. However, if your bleach is greater than 50%, you will be forced to go into modeling non-linear kinetics and exponential kinetics, which is something which is a little more difficult. And if someone is interested in that, I can cover that later. Right now, I'm considering the ideal case where your bleaching is still in the linear range. Okay. So now moving on to the test sample. If everyone can open the test sample. I've already marked the ROI. If people are interested, they can mark it themselves as well. If you can continue with all of the procedure that I told you till now, which is image, stats, and plot your axis profile, what you'll see is a wonderful recovery plot over here. And it's essential to have at least 5 to 10 frames before you start your bleach so that you can normalize effectively. You can average out that intensity and see where you start from. So you have to take 5 to 10 frames before you photo bleach your region of interest. So as you can see, I've taken 5 frames over here. And after a time, there is dramatic loss in fluorescence. So now if I normalize this to 0 again, like we did previously, you can see that the intensity lost is around 75%. And then it recovers back from that region. Okay. So now that we have this curve, we will again, like previously, list out the intensities per unit time. So here we can see the intensities per unit time. And what I had previously done is to manually, in the metadata of the image, I had already set the time scale of my movie. So this is not essential. All microscopes may not automatically do this. If your microscope does not support automatic output of the time, you'll have to manually do this in the Excel software. So thankfully for us, we already have it, which is the exact time as well as the intensity for the frame. Now we do the same thing, which is just select all the values, copy paste it in Excel. So now we can have our test data set over here. The first thing that we have to do is to normalize it like we did previously. So normalization here is a little more detailed, because the first thing you do is average out all the pre-bleach numbers, because we have to be sure of where our pre-bleach intensity actually was. So for this, we just put equal to and we put average, and you average the first 6, in this case, pre-bleach columns. There's a question, if we see for the recovery, it is not coming back even 50%. Why is it so? So there could be multiple reasons. The first one is that there is some bound fraction. So not all fluorescence recovers in the region of interest. And it's, let's say, bound to some particular component, let's say a vesicle, or let's say a membraneless organelle. Let's say if you're bleaching within the nucleus, is a bad or rapid recovery from the cytosol. So those are common reasons why we may not see complete recovery. Okay. So moving on to the analysis, you average out the first 6 values because it's imperative that we know where our intensities are starting from. The second thing to do is to put, again, is equal to this, which is your intensity value, divided by your normalization value. And like previously, we will put a dollar sign before and after. So it should read as dollar, whatever letter, in this case I, $1. And you press enter. You'll have this intensity normalized again. And if you just double click the bottom right of that cell, that intensity will be carried forward till the end of the data set. Now, the final thing that we have to do is a very simple thing. We have to reorient this time scale, time series, so that 0 comes as the first post-bleach level. Because all the equations that we discussed till now assume that you are starting from a low intensity and rising up. They don't assume you're starting from 1 and then go down and then come back. The only reason the pre-bleach intensities are required is for correct normalization and to see how much recovery is happening. After we have normalized, the pre-bleach values lose their significance and do not have to be used further. So we have to reorient this time series so that the first post-bleach frame starts from 0. And this is simple enough to do, since we just have to put a simple equation, which is put equal to this time series minus 3, since 3 is the first frame. And you just double click it so that now this is reoriented such that your first post bleach is 0 seconds. Now, if your microscope does not support time series that is exported as metadata, you'll have to manually put whatever frame rate it is. So as an example, so this suggests that it was 2 frames per second, since it takes 2 frames in 1 second. If it was 3 frames per second, it should be the difference between two cells should be 0.33. If it's 4 frames per second, it should be 0.25. So that's something that you'll have to manually then do. Once you have these data sets, what you do once you have this data set, you just copy paste it in another region. Just copy paste the values. For some reason, zeros also came here. Yeah. So just copy paste. Oops, sorry. Second. Yeah. So you can copy paste the values, and now you'll get 2 columns like so. One is time, and the second is the normalized intensity of your recovery profile. Can you just repeat this time calculation? Oh, yes, yes. So what you do once you have this this time series, you just put the equation, which is equal to your time cell minus whatever value is in your first post-bleach frame. So in our case, the first post-bleach frame has a value of 3. So you just put 3. And then you iterate it over all time values. That's it.
Can you help me how to copy that whole time frame?
Oh, yeah. So you just, okay, you press on that cell, you press shift and you double click the bottom line of that cell. It will automatically go down and select the entire row. Like that. Did you see? You just have to select the cell, press shift and double click the bottom line of this cell. That's it.
To paste it because it's coming -3 wherever I'm pasting.
Sorry.
It to another cell, it's coming -3.
Yeah, just remove those -3. For some reason, it iterated even further than just
No, everything is coming -3.
delete those minus. Oh, okay. So what you did, when you
Everything
press
is coming
equal to,
-3, whatever I'm pasting.
yeah. So what you have to do is you have to put equal to. You have to select the cell first, which is the time cell. As an example, here it would be F1, and then F1 -3. You have to press. And once you do that, then you can drag this and it will auto complete. Is that clear?
Yeah. And how have you pasted this into the J cell from?
So what you do is you first select all of the regions. You press Control C to copy, and you just do a special paste, which is paste here on the top left, and go to category somewhere. Because it which will be -3. So what you have to do is paste the values, which will actually paste as you see. Yeah. So once you have this, now what we are doing -3. So Tejal, there could be 2 errors. One is that you didn't specify the cell. The second, Akshay answered. So once you have these numbers, now we can correct for acquisition. The density of the amount of photo bleaching. Okay, can everyone hear me or is my voice breaking? There's someone who has. Okay, I guess my voice is. Just give me a second, I'll reconnect to Wi-Fi. Okay, can everyone hear me now? Is it better?
Yes, we can hear you. Yeah, it's
Yes,
better
okay,
now.
cool. Yeah. So once you have this time series, what you have to do is to correct for acquisition bleaching. Since we have, let's say, 30% loss in intensity without you shining the laser and high intensity laser and doing anything, we have to somehow correct for it. And since we were still in the linear range from the movie that I showed you before, a simple thing that we can do is just put equal to, okay, before that, we'll have to do one more thing, which I forgot to tell you. You will have to make a time series which iterates with the row number. So you just put 0, 1, 2 and double click it so that it auto completes. This will be relevant later where we're correcting for the intensity. I'll show you why. So you just put equal to this intensity plus this intensity. Sorry. So this will be minus this intensity because this is a negative. So, okay, I'll just go over this once again because this can get confusing. So when we plotted the acquisition bleaching, you can see that the slope is negative because the intensity was reduced. So to correct for it, we have to somehow add this slope value to this curve such that the intensity gets normalized again. So now this is not something that you'll have to do for your experiments, but it's just something that I'm going to show you of why normalization of intensity is so important. Okay, so I'm going to use the same equation, which is to correct for acquisition bleaching as I was showing later, which is just to put equal to your intensity minus bracket, you put this intensity in dollar signs. So $E, $1 multiplied by your frame number. So your this iteration 0, 1, 2, 3 is basically the frame number of your intensity. If you just put this equation, this will now correct for any loss of intensity over time. So if you just do the same equation over the entire column and now plot the data. So this is your original normalized intensity without correcting for photo bleaching. Once you correct for photo bleaching, now you can see all of your intensities are around 1, which is there is no loss in photo bleaching over time now. Why you see these up and down steps is because of noise in the system. Okay, Kajil, this is not something that you need to do. It's just something to understand of why acquisition bleaching is important. So I'll just go over the explanation again. If you have any intensity like this, acquisition bleaching, which is reducing with time, you have to correct for this loss in intensity somehow. Since we have this slope value, we can use the value for intensity loss over time. And since it's linear, we just need to add this number sequentially to each one of these rows. That's it. So that's what we're going to do. So we're just going to put equal to this intensity value minus, and why we put minus is because this slope value is negative, and this slope value is something we have to subtract. So in short, we have to add a positive integer to this value to bring it up to 1. That's why we put a subtraction sign. So then we put bracket, this normalization coefficient, which is followed by dollar signs, multiplied by the frame. What this frame does is basically, sequentially adds this number every step you take. So this number will be now 0.00131 more than this. This will be 0.00131 more than this. Eventually, it's the entire column. And once you do this, your intensities now hover around the intensity 1, as can be seen by this plot here. So now your image has been successfully normalized for acquisition bleaching. So this same thing, you don't have to do this for your data. It's only for your understanding of why correcting for acquisition bleaching is important. The same thing is what we're going to do here. So just copy pasting this formula where you put equal to M7, which is your first post-bleach frame of interest, subtracting by the the acquisition bleaching coefficient, multiplied by your frame rate, the frame number, which is here 0. And you press enter, and you'll have some value. And you just so
Hello, Vidur. Hello, Vidur. Your voice is breaking.
now
Yeah, maybe you can switch off your video so that it will be better.
am I audible now?
Yes, you're audible. If you switch off your video, your voice may get better.
Oh, okay. Switch off my video. Okay, so now that we have these columns, what we will do is just copy paste them. You just copy them. So calculating using normalized value or the actual value. It's always calculated using normalized value because the actual will check on the conditions of the scope, the intensity of fluorescence. This analysis will always be done on normalized values. Now, what you have to think, just copy paste values and go back to image. Press image.
Hello, Vidur. Your voice is still breaking.
Is this better now?
Hello, Vidur. Your voice is still breaking.
Is it better now? Is it still breaking?
Now it is better.
Okay.
Yeah, you can
So
continue.
I'll continue. Yeah. So when you now going back to Fiji or Image, what you have to go to Analyze, Tools, and then press this curve fitting bar. So everyone just follow the steps, Analyze, Tools, curve fitting. Once you press this, you just copy paste the values that you copied from Excel, which is your normalized intensity for both normalized to the pre-bleach values as well as the amount of acquisition bleaching. So you'll get values here. Now you go back to the data set that I had sent you, and there is this one text file called equation.txt. And you copy paste this equation. You copy it and you go back to Fiji curve fitting window. Okay, I'll repeat the last 2-3 steps. You first go to Analyze, you go to Tools and you press curve fitting. Once you have this curve fitting window open, you'll see something like this. You just paste your data over here. Once you paste the data over here, you go to the equation file that I had sent you. It must be attached in the unzipped file that Akshay had sent you earlier. And copy paste that equation because that's going to be relevant now. And what you do is to go press on this straight line button and press user defined and press fit. It will ask you for a formula. You just have to copy paste the formula that I have given you before. I'll explain why this formula is as such in a while. But once you press OK, it will automatically fit your data and give you the constraints that are required. Okay.
Can you please repeat it again?
Yeah, from what step should I repeat it?
Copying this data on the curve fitter and after copying equation text.
Yeah. So once you copy paste the data in curve fitter, what you have to do is you press this drop down menu. It has many options here. What you have to do is press on user defined and press fit. It will ask for a formula. Just put this formula, y is equal to a into 1 minus what math.exp. So basically, I'll explain this formula right now. A is your mobile fraction. Math.exp replicates the exponential e, and this is e to the power minus b into x. X being your time and b being your half-life tau. And C is just a parameter that indicates that you're not starting from 0, you're starting from some arbitrary value like 300, which is, since we don't photo bleach it completely till 0. And you just press OK. And your Fiji will automatically fit it. So it just so happens that it's not the best fitting here. You can see that there is structural errors. It's not completely random across the thing because my data suggests that there is a 2 exponential fitting here. But this is a raw thing that everyone can try their own data set. Okay, did everyone get this curve?
Whether one participant is saying it's showing bad formula in my Fiji. And one person is saying, can you please explain it once more?
So for the bad formula in Fiji, I'm not sure what is going on. I'll have to see your screen. So I'll suggest go into a breakout room where I can check your screen and see what formula it is there. Which we can do in about a minute or so. And the second, which is, can you please explain it once more? Where do you want me to explain from? Can you just let me know where does your confusion start from? From where you got the formula in Fiji. Okay. So, okay. So, to get the curve fitting window, I guess you all are on the same page, analyze tools and curve fitting. Once you have the curve fitting window, this curve fitting window will originally have an option called straight line. However, we are not interested in the straight line option. What we're interested in is in the recovery, the frac recovery function. So for that, what we have to do is to put a user defined formula. So I had shown you previously that the recovery profile is basically modeled by this X. I'll just go back to this slide so it's a little easier. So I had already shown you that you can have a sort of parametric estimation of what the recovery profile looks like based on this formula, which is basically A, the mobile fraction, multiplied by 1 minus tau multiplied by t raised to the exponential e. So this was a formula that we started with, which I told you that you can look at this resource to derive the formula. Now, since we know this formula, we have to do a simple thing of putting this in a way that Fiji can understand it. So how Fiji understands it is by putting y, which is your intensity parameter, equal to, so y is basically all of these second column values, which is equal to a, which is a constant that needs to be fit, which is your mobile fraction, multiplied by 1 minus e raised to the power. So how e raised to power is read by Fiji is by putting math.exp. Raised to the power minus b into x. This minus b is none other than the tau variable that I had told. And x is your time variable, which is 0.511. And so this entire formula till here is exactly what I showed you in the presentation. The only reason why we start from plus c is because the equation assumes we start from 0. However, since we can't have 100% photo bleaching, we start from some, let's say, 2.24 intensity. That's why plus c is required. Because this is an offset, which suggests that we start from this 0.24. Once we have this equation, you just press OK, and Fiji will do the rest and fit the best equation for you. This A is your mobile fraction. To calculate your half-life, you just have to have ln 2, which is 0.693 divided by B, which is 0.03, and that will be your half-life, which will be somewhere around 20 seconds. And finally, T is the offset. It just suggests that your y-axis starts from 0.0304. This is not the best fitting in the world because this is the imaging that I've done does not have a single exponential associated. But this may be true for your particular experiments. Am I clear? I hope I'm clear now. If anyone has a question, they can tell me. Otherwise, I'm going to now stop my session. There is another sample 2 which I have associated, which has two different fluorophores, unconverted, which is a kinesin motor I'm interested in, tagged to GFP, as well as a soluble M-cherry control. And in your spare time, you can try assessing the recovery rates of both protein tagged GFP as well as a soluble M-cherry to see how differences can be quantified. But that's something you can do on your own after you know this. The second thing is, what is half-life in the equation? Okay, just to go back to my presentation. Here, you fit using a constant called tau. This tau is related to half-life by this equation, which is half-life is equal to natural log of 2 divided by tau. So natural log of 2 is around 0.693 divided by tau. That will give you a half-life. The second question is, the offset is what you set on the microscope while acquiring, or is it something related to Fiji itself? Okay, the offset is a constant that is being optimized here. That's not something that you set. It does not matter what that value is till the fact that you keep what I've said till now in mind, which is you have to have 80% photo bleaching, around 80% photo bleaching. So that's something that you have to have in mind. Apart from that, you don't have to care about the actual offset value once you get it. That's something that Fiji is itself optimizing to give you the best fit. Okay. Am I clear? Yeah. So I'm going to stop my screen share if no one has any other questions, and I'm going to just go to a breakout room to see if anyone has a bad formula in Fiji, as was being there for Alice.
Yes, whether just before
Anyone
that,
has any questions?
before we go for the doubt solving, maybe some of the participants can volunteer to show their graph. If anybody has
Oh yeah,
went to
sure,
the
sure.
final level, yeah, then we will know if how much, yeah.
Yeah, that's
Can
fine.
someone please share their screen and show the graph if you have got it?
Yeah, that's great.
Yeah, so we will make a breakout session now and add the participants who have doubts and yeah, Bidur in that session.
Yeah. Okay, I guess all questions like people can ask in the breakout session, and Shravanti can load her screen and start. Or I think we're running out of time because
Yeah, I guess we only have an
okay, there was
hour.
yeah.
Everyone. So the breakout session is created. I'm given permission for everyone. Whoever wants to go there can directly click on join and go there. But in the meantime, Shravanti, you can start.
Yeah, I had to rejoin because of network issues. Could you just make me the co-host when after the, I mean, by before this ends, so that in case I have to make a breakout room, I can. Otherwise, I'm okay.
That's fine.
Yeah, that's okay. I guess I will